All right, I'll be going over part one of chapter 13 in this video, what we would have been going over this week before Thanksgiving break. And then when we get back from Thanksgiving break, I'll go over the other half of 13 and any questions you guys have from part one since we weren't able to meet in person. So, chapter 13 is getting into the gross anatomy of the brain, and then we kind of finish off with the cranial nerves, which are something we definitely want to look at for knowing, different functions of these nerves that branch from the brain. So, we'll start right into it. So, there are four main regions to the brain from a gross anatomy perspective. The largest region by far is cerebrum. And cerebrum is separated, you might see, in two hemispheres, left and right. But there's also separations by lobe, and there are 5 lobes that we will talk about as far as the brain goes for cerebrum. And again, I'll put my mouse over it. This is cerebrum, very large portion of the brain. If we cut the brain in half, we can see the innermost portion, which is diencephalon right here. And it encompasses a few structures. And then we have the brain stem, okay, coming down here. And we have cerebellum, which is sitting below the occipital lobe. So those are the main regions that make up the brain. There's a few features that we can use to describe things. The outer surface, which is used to describe cerebrum, we have these two terms, gyri and sulci, those are plurals. All of these ridges are convolutions, right? The meat part of cerebrum is called a gyrus, or multiple would be gyri. And then all of the shallow grooves in between, right, that create these separations, those shallow depressions or grooves are called sulci. We will look at an additional type of groove in the brain that typically we're going to see when we separate the two hemispheres of cerebrum. This is a much deeper cut, and we would call that a fissure. We can even see, a few examples. Right here, if we cut the brain in half, there would be a fissure in between these two hemispheres. Additionally, there's some new directional anatomy terms that can be used, especially when describing the brain. So instead of anterior or posterior, we could use the term rostral for the brain, rostral meaning in the front, anterior, and caudal meaning the back, posterior. So if we take apart the brain and we want to look at what components are made up for it, we typically call those components gray matter and white matter. And in most organs, we usually have a name for the components on the outside versus the inside. The outer layer is usually called the cortex for most organs, and the innermost layer is called the medulla. So the cortex of cerebrum, and we can kind of see here it's a little bit darker in color versus the middle medulla. The outer portion, the cortex of cerebrum, is gray matter. And it is darker because it contains all of the neuron cell bodies, their dendrites, and any unmyelinated axons if those neurons are present. And so what we're truly seeing staining, especially when we look at microscope slides, is going to be the cell bodies, and they'll stain darker, and we call them gray matter, making up the cerebellar cortex. In the center, which we would call the medulla here, we have white matter, and all the white matter is the axons coming from the neurons from gray matter. And those axons are myelinated with those myelin sheaths, which have a white waxy appearance, which is why we call it white matter. And bundles of white matter, okay, these myelinated axons we call tracts. Additional tracts come together and eventually form nerves when we enter the PNS. So let's take a look first, okay, at some of the support structures for the brain. This will be included for the spinal cord, but we'll talk about the spinal cord in chapter 14. So there's a few structures that protect or support the brain. One, of course, we've already learned is the skull. The skull is going to house different structures of the brain, and it is a primary mode of protection. We also have protective membranes around the brain called meninges, and we'll get into the layers. Cerebral spinal fluid is present. It provides cushioning and nourishment in the brain. And of course, we have the blood-brain barrier, which is bringing nutrients to all those neurons. And we already learned a glial cell, which is part of the blood-brain barrier, which is the astrocyte. So let's look at our meninges, and we'll just refer to these as cranial meninges, okay? There are 3 of them, and they are protective for the brain, okay? They're going to be found underneath the skull tissue, okay? And just to orient you to this picture, because you have this in your lab as well, this is the skull right here, right? We can see compact and spongy bone, and then underneath here, we have the brain, right? We can see the gray matter and the white matter, and we're going to be looking at the layers in between. It looks like there's more than 3 layers here, but that's kind of false because there is just 3 of those meningeal layers, but there's a lot of spaces between the layers, around the layers, that give it the impression that it's more. Our layers are called pia mater, arachnoid matter, and dura mater. Pia mater is the deepest. It's going to actually touch the brain. Arachnoid matter is in the center. And then dura mater is the one that is interacting more with the skull bone on the outer portion. So we can kind of take a look. We can look at pia mater first. This is this very thin pink line, okay, that we see kind of going over here on top of the brain, interacting, and it's just a really thin layer of areolar connective tissue. If we switch over, we can start to see we've got a lot more structure to the other layers, one of which is arachnoid matter. There are these trabeculae, these branches that are occurring in arachnoid matter, and they're called arachnoid trabeculae, and they look like spider webs, which is where we get the name arachnoid matter, okay? And there is spaces, okay, in here, giving it a much, more complex impression, right? One of those spaces is the subarachnoid space, okay? Subarachnoid space can fill with cerebral spinal fluid. And then additionally, there's going to be a subdural space also interacting with arachnoid matter. And the subdural space is where we can have blood go, okay, if a vein ruptures. That is all before we get to the outermost layer of dura matter. Dura matter itself, okay, is tough outer membrane. It's dense irregular tissue. There is a meningeal layer, which is kind of deeper. And then there is an periosteal layer. It has that word ost in it, meaning it's the part that actually touches the surface of the skull bones. And there's also a space present there as well. We'll take a look more at this more deeply in other pictures, especially in the lab where you can see those structures. But again, we're just focusing on the meninges, these 3 layers. We do have a clinical view for this. These are epidural and subdural hematomas. These are referring to the spaces. Epidural would be outside the dura mater. Subdural would be below. A hematoma is a pool of blood that can form. So an epidural hematoma is one that forms in the epidural space, okay, outside of dura mater, usually due to some sort of blunt force trauma to the head, okay? And this can lead to severe neurological issues and even death if the brain bleed cannot be removed. And the blood will need to be drained from this type. An additional type of hematoma could be a subdural one. This would be below the dura mater. Typically, if you have ruptured veins, okay, this is also from blunt force trauma to the head, although it has more of a rotational fashion. This can compress brain tissue, and this blood pooling will fill more slowly. The subdural space is meant to contain blood, and this will occur more slowly. You will still need to have this blood removed. So these are hematomas, brain bleeds. There's some other clinical views involving the meninges. One is meningitis, right, which probably most of us have heard of. Itis is just a prefix, a suffix, excuse me, that means inflammation of the meninges. And typically, this inflammation is going to be caused by some sort of infection to these protective layers around the brain and spinal cord. Symptoms include what we would typically see with viral or bacterial infections: fever, headache, vomiting. You may have stiffness of the neck, especially if it's occurring in that area. And typically, it's going to be pain in the back of the neck. Again, meningitis is a very serious condition. It can lead to brain damage. It can lead to further infections in the brain and even death if untreated. And typically, bacterial meningitis is seen to have more severe symptoms. The last kind of clinical view involving the brain and the meninges is encephalitis, inflammation of the brain itself, most often from viral infections that are able to spread to that tissue. And some symptoms include drowsiness, fever, headache, neck pain. And again, if encephalitis is not taken care of with antivirals, okay, and therapy, this is also a very serious medical condition as well. So let's talk about some other structures in the brain, okay? Aside from the protective ones, we mentioned that cerebral spinal fluid is also protective. It provides nourishment as well as cushion for the brain tissues. And the ventricles are really great for housing cerebral spinal fluid because they are hollow cavities within the brain. And they are just colored blue here to show cerebral spinal fluid having kind of a full flow through here. There's no stops in these connections. This will flow all the way through all these ventricles that are connected together continuously until it reaches the spinal cord. And again, we know that our ventricles are lined with one of our glial cells, which are ependymal cells. So there are 4 ventricles, okay? And then there are a few structures that we also look at as well. There's 2 lateral ventricles, right? One in one hemisphere, one in the other. They're also often called the lateral horns, okay? And they contain a lot of cerebral spinal fluid. They are connected right here by interventricular foramen to the 3rd ventricle, which is right around diencephalon. And then they are connected to 4th ventricle on the brainstem through the cerebral aqueduct. This is continuous flow of cerebral spinal fluid, which is nutritive and cushioning for the brain tissues. And then eventually, after you get to 4th ventricle, this will flow, okay, into the spinal cord. So there is continuous flow of cerebral spinal fluid, and the part of the spinal cord where cerebral spinal fluid flows is the central canal. So just to talk a bit about CSF, okay, which is our abbreviation for cerebral spinal fluid, it should be clear and colorless, okay? When people take cerebral spinal fluid and it has a cloudy or colored appearance, it's usually due to infection. And our cerebral spinal fluid circulates in our ventricles as well as in the subarachnoid space, so around arachnoid matter. There's a few functions. It provides buoyancy, reduces the weight of the brain, its protection as a liquid cushion, and environmental stability by transporting nutrients and wastes for the neurons. So cerebral spinal fluid is very good for our brain tissues. So let's get into the brain tissues. So we'll start with cerebrum, okay? And cerebrum, again, is this very large portion of the brain. It's separated into 2 hemispheres, right and left, but it's also designated into lobes, which have their own complex intellectual functions. We're going to go into all of those aspects for these. Collectively, cerebrum is your center for intelligence and reasoning, thought, memory, and judgment, muscle control, conscious perception of senses. There's a lot going on here, and we're going to look at these functions by lobe, okay? So we'll start here just by looking at just, some of the designations for the cerebral hemispheres. All right, so we've got left and right here making up cerebrum, and they've even kind of separated in this picture, kind of the pictures of the lobes that are going on here. And right down the center, separating the 2 hemispheres, is longitudinal fissure, right? It's one of our deeper depressions. And again, surrounded by all these shallow sulci. And then we actually know that these 2 hemispheres communicate with each other. And this is through a tract. A tract is axons, right? and white matter, basically connecting these two together so they can communicate. And that connection is called corpus callosum. This allows them to send signals to each other. We do know that for a lot of different functions, we have a kind of crossover here. So when they are connected by corpus callosum, each hemisphere interacts with the opposite side of the body, which we call contralateralization. For example, the left hemisphere will receive signals from the right side of the body and send motor signals to the right side of the body. And that's kind of how they work. So let's look at some of these lobes here, okay? There are 5. You might notice that you're only learning 4 in lab. We have the frontal lobe, parietal lobe, occipital lobe, and temporal lobe, so named for the bones that you learned that protect them. And then in the center, okay, surrounding diencephalon, you have insula, which is also called the insular lobe. So let's look at some of the functionality for cerebrum. And probably for today's lecture, I will just be going over cerebrum, and then we'll finish chapter 13 on after Thanksgiving break. So for cerebrum, let's start with the frontal lobe. We have a lot of names actually for the gyri, okay, that are present here. You don't have to memorize the names of the gyri. Don't worry about that for this. But we do want to know what kind of separates out frontal lobe and some of the functional areas. So on the frontal lobe here, we can see it's separated from the parietal lobe with a sulcus called central sulcus. And then technically this gyrus here, which is red, is called precentral gyrus because it's before the central sulcus, and it controls movement because it contains our primary motor cortex. So essentially, and there'll be a table for this, essentially the frontal lobe is controlling a lot of motor function. We also have a lateral sulcus, as you can kind of see right here. Your lateral sulcus is going to separate your frontal lobe from your temporal lobe, okay? And we're going to see what the temporal lobe has to offer, okay? But there's going to be a lot of functionality here that we're going to see. Motor control, like we just mentioned, concentration, verbal communication, decision-making, planning, your personality, all existing in your frontal lobe. Let's look a bit deeper, okay? We know we have a motor area here, okay? But there's a few other areas also present on the frontal lobe, okay? One of which is your motor speech area called Broca's area. It is considered inferiolateral portion of the left lobe in most people, and it controls movements needed for speech, vocalization. We have a frontal eye field as well, okay? This is found on middle frontal gyrus, okay? It regulates your eye movements needed for reading and binocular vision. A lot of this has to do with motor functions for things. We also have a premotor cortex, okay, located anterior, okay, and our premotor cortex coordinates learned skills and activities, okay? So anything that is going to come from a muscle memory, okay, of how to do something is going to be present on your frontal lobe. We have a prefrontal cortex as well, okay? And the prefrontal cortex here, this is the part that gives you your complex thought, your judgment, personality, planning, and decision making. And this is still developing in adolescence until you reach about the age of 25. So lots of areas on frontal lobe, and I promise there's going to be a picture where we're going to kind of associate all of this. For parietal lobe, okay, this is where we have the post-central gyrus, right? Because this is our central sulcus. Post-central gyrus is behind in the purple here. This is your primary somatosensory cortex. This allows you to understand by touch the shape and texture of objects. So say, you know, I gave you guys a mystery bag full of items and you needed to, if you put your hand in and you touched it, you would know what the item is, not by sight, but by touch, and that is coming from your somatosensory cortex. For the temporal lobe that we have here, separated by this lateral sulcus here, we have a few functional areas. We have our primary auditory cortex and auditory association area for hearing. It's not surprising since we know the temporal bone has your ear canal in it. And we also have areas for smell, your primary olfactory cortex. Looking at a few other areas, okay, especially associated here with our parietal and temporal lobes, we have some functional areas. One is Wernicke's area, okay, which we can kind of see spreading in kind of both areas here for the lobes, okay. And typically, this one is going to be involved for your language comprehension. So when people are speaking to you, your ability to process and associate that with certain things. Occipital lobe is primarily for vision. So functions for vision and visual memories. We would call these our primary visual cortex and our visual association area. You can think about it a lot. We see this in the sport of hockey. When people get hit on the back of the head, they can end up with double vision or maybe a loss of vision temporarily. This is due to damage to their occipital lobe. The last lobe is insula, which is internal. This is a very small lobe, okay, and it functions for memory and your sense of taste, which is called your primary gustatory cortex. I will mention in here, I like these terms, okay, because they are the more scientific way of speaking about your special senses, which is going to be vision, smell, hearing, taste, okay, those types of things. And so taste is referred to as gustatory, just like we might look at, smell is olfactory, hearing is auditory. Okay, so a few of those functions. And this is the summary table that I would have you guys look at, okay, especially for learning some of these areas. So we've got the frontal lobe, temporal, parietal, and insula. And again, it talks about their functions here, what their functional areas are called, and then in general, what the summary of their functions are. So if things happen to the frontal lobe, right, it can affect your motor function, your personality, your ability to think and plan. If things happen to the temporal lobe, it affects your hearing and smell. Parietal lobe has to do with touch and identification of objects by touch. Occipital lobe is vision, and insula is taste. So these are kind of our lobe summary areas for what each lobe of the brain does. And depending upon what happens to a patient due to an injury or an infection, we can tell what part of the brain has been affected. So we'll stop here. And then I will continue after Thanksgiving break with diencephalon and the rest of chapter 13.